Hi, my name is Katherine Rice. I'm the Applications and Market Development Manager here at CAMECA in Madison, Wisconsin, and I'm very pleased to present a very short tutorial on atom probe tomography and its application to semiconductors, which might be relevant to the Electronic Failure Device Analysis Society. Let's jump in. So just a little bit about me and my involvement with the society. I received my bachelor's in chemical engineering at the University of Maine and my PhD at the University of Colorado Boulder. I was able to do an NRC postdoctoral fellowship at the National Institute of Standards and Technology, also in Boulder, working on electron microscopy techniques. I've been at CAMECA just under 11 years now, and I'm the Application and Market Development Manager here in Madison. We do customer demos, we do training, and we find new ways to use the atom probe for new applications. As for my involvement with the Electronic Device Failure Analysis Society, I was pleased to be a proceedings author in 2020 on automated specimen preparation of atom probe tomography samples, and was lucky enough to do a tutorial at the 2024 ISFA meeting. I look forward to also being a tutorial presenter at the 2025 meeting. So in Atom Probe, we start with a needle-shaped sample. This is extremely small. The end radius of this needle is somewhere between 50 and 100 nanometers. We can then apply a standing voltage to the specimen so that the ions are almost but not quite ready to evaporate from the surface. We then give the sample a little bit of extra energy in the form of more voltage or a thermal pulse from a laser. That essentially causes the ions on the very surface of the sample to evaporate one at a time. Now, as we all remember from high school chemistry, every element weighs a different amount. Your carbon weighs a different amount from your gold, for example. And so the time it takes for an element to evaporate from the sample and reach the detector, that gives us a time of flight and can be correlated to the mass and therefore chemical identity of that ion. From where that ion impacts on our position-sensitive detector, we can essentially back calculate where it came from on the initial sample surface. So when all is said and done, we combine that mass information and position information, and we have a 3D reconstruction of up to 50% of the ions in your sample. Now, once you have that volume, the possibilities are really endless. You can look at things like concentration profiles, local regions of your data set, and so on. And it's important to remember that this is a truly three-dimensional technique. A lot of times in our microscopy world, we're working with two-dimensional images. In this case, we have 3D information that contains both compositional and positional information. So this can be very valuable for materials analysis of all types. Now, one of the first questions that I'm always asked is, how do we actually get a sample in the shape of a needle? And we do that usually with a dual beam fib. I know the failure analysis community is very well familiar with FIB, so it should be no problem for someone who's very competent in TEM samples to make an atom probe sample. We start by identifying the region of interest and protecting that with a little bit of platinum or whatever capping material that you have in your gas injection system. We can then use the ion beam to make cuts on three sides, so we have kind of a cantilevered wedge still attached to our surface. We then bring in a micro manipulator needle and do a little bit of welding of that needle onto the surface of our sample and cut the remaining side free. We can then move this wedge over to a flat-topped silicon mounting substrate and essentially weld a little piece of that on and cut multiple samples from that wedge and get a little bit of tacking material on the other side. Now, the sample does not yet look like a needle, so there's one more step that we do, which is called annular milling. Essentially, you're using the ion beam in your fib, much like a pencil sharpener, to kind of whittle away material so that you have a needle. And this allows the region of interest to be really targeted within a few nanometers. Especially with the technology development on the fib side, we're able to target those regions of interest within about 30 nanometers. And this is really helpful if you're trying to target increasingly smaller electronic devices. Now, for advanced sample prep, sometimes we need to do that with semiconductor materials. We can also prepare things in a planar or cross-section view and also approach the sample from the backside if we are trying to avoid different regions in our sample. So there's a lot of options on sample prep, but generally the way these are made for electronic devices is with a dual beam FIB. The basic unit of data that we're getting from the Atom Probe is a time of flight mass spectrum. Now here I'm showing a piece of that mass spectrum from a semiconductor device. What we can do here is compare the peaks in our data set, is our mass spectrum, to what is theoretically expected for an isotopic distribution of a particular element or compound, in this case hafnium oxide. If our peaks are a good match to that ratio, we can have a high degree of confidence in assigning the masses that fall within that region to a particular element. Now, one great thing about Atom Probe is that it's equally sensitive to all elements in the periodic table, meaning we can detect light elements like boron just as easily as we can detect heavy elements like platinum. And this is kind of rare in the material analysis techniques when you think about it, because a lot of techniques that rely on X-rays have difficulties detecting light elements. So what we can do is go through our mass spectrum, compare that theoretical distribution of isotope ratios to what we have in our data, and assign those masses to a particular element. We're then going to color those ions by their chemical identity and assign that to our 3D volume. So now we know what those elements are and where they are in our data set. Now, once we have those assignments and have gone through our mass spectrum, we can do things like create what we call a virtual TEM lamella by selecting a slice of data and displaying the elements individually so we can see their distribution. Here we've broken out the titanium nitride ions and our silicon and oxygen ions, as well as our cobalt, germanium, and boron ions. So this allows us to look through an equal volume of data instead of looking through that cone-shaped material and see where these ions are more or less concentrated. Now with true three-dimensional data, many times you want to separate regions in your data set. Something you can't do that easily in a 2D image, for example. So one of the things that we can do is choose criteria that we want to apply as to how to define regions in our data set. In this case, we use a technique that we call an isoconcentration or an isodensity surface. So here we have a full data set. We can define regions of our data set that meet a particular concentration. In this case, I've chosen a 95% cobalt criteria. So what that means is that the ions within that surface have a concentration greater than 95% cobalt, and on one side of the surface, they have a concentration less than 95% cobalt. And that allows us to really isolate these from the integrated bulk of the data. Now, here we've selected one of those regions. I've chosen the top contact that kind of looks like a brain. And the other thing that we can do is display the mass spectrum of just that region separately from the rest of the data set. We can then analyze that mass spectrum for a bulk composition of just that top cobalt piece. And that's going to allow us to find concentrations that aren't necessarily mixed with the rest of the data set. One of the things that we're often interested in is how the concentration changes from one region of the data to another. Maybe you're looking for something like a diffusion profile. And there's a few ways that we can actually do this. One is to create a profile. Here we have a cylinder that's going through kind of a particle that's highlighted by one of those surfaces that we just talked about. We can create a profile by taking concentration measurements at each given point in that cylinder, kind of slicing that up like a slice of bread to get measurements from the matrix and then the particle and then back to the matrix. And we can also look at things radially. So here we have a particle that's defined by a certain concentration criteria, and we can look at how the concentration changes going from that interface out into the matrix, but also inside that matrix as we approach the center of that particle. And these are really useful to find out how gradients might be occurring in your data set. So the basic workflow of Atom Probe might look something like this. We would have a sample, maybe that's been depackaged or deprocessed so that the region of interest is relatively close to the surface. Maybe it's something like a short loop wafer. We can then do our sample preparation, which we talked about in the fib, for a basic top-down style lift-out. This would take maybe 4 hours to create 6 samples or so. Sometimes if we're doing some of those more advanced specimen preparation techniques, like approaching this from the backside up, this might take something like 12 hours to get 6 samples. We can then acquire the data in the Atom Probe, and this is totally variable depending on your sample survivability, where your region of interest is, and what your experimental goals are. But just to give you an idea, it can be anywhere from 30 minutes to a long data set might go overnight. Then to reconstruct the data, that's applying those mass assignments to your position data. This could take anywhere from 5 minutes to an hour if you're being very careful. And then the analysis side is totally variable on how long that takes depending on what the goals are of your analysis as well. So what kinds of things can we analyze with this, and what are the general applications? And for SEMI, this has been an area that's been growing, and it's really exciting to see the data that's coming out of industry right now. We can do things like look at entire semiconductor devices. And this is really useful if you need to see the interplay between what these different structures are doing, compositional gradients, things like that. So we can look at things like entire transistors, we can look at contacts, we can look at back end of line. We can do things like look at clustering problems. What is clustering and where? And we'll see an example of this here in a little bit, but that might tell you things like how many of your dopants are actually in clusters. And we can look at things like profiles. How is your concentration changing from one region to another? Atom Probe really works best with samples that are solid materials, so not voided, and at least a little bit conductive. So typical applications are metallurgy, semiconductors, geology, and not too many applications at this point in time in the biological sciences. We can take a quick look at how Atom Probe Tomography compares to some other typical materials analysis techniques used by the semiconductor industry. Here on both y-axes, we have a detection range, that's a sensitivity or concentration that we can collect. And on our x-axis, we have a lateral resolution. That's basically how many ions or what volume we need to measure to get that particular concentration. We see that Atom Probe Tomography occupies a place in this materials analysis landscape that's really difficult for other techniques to reach. Now, ultimately, the sensitivity of any of these techniques is going to depend on how many atoms are being sampled. For Atom Probe, that's not always determined by the analyst. A lot of times the sample determines how many ions you collect. At some point, samples can't undergo the stress of this process of evaporation and they break or fracture. Now, how does that maybe relate to some semiconductor problems? We can take a look at an example published in 2018 by Global Foundries of a FinFET device. You'll note that there's no scale bar on this image. I think it's unlikely that it would be published if those dimensions were revealed. But I can tell you that typical device dimensions and concentrations for the semiconductor industry fall very well within the typical analysis space measured by Atom Probe Tomography. And that makes this technique really valuable for failure analysis problems. Let's take a look at some example data from the Atom Probe that might be relevant for electronic device failure analysts. Here on the left, we have an SEM image of an off-the-shelf semiconductor chip. This is a Snapdragon cell phone modem chip, kind of a later technology node. In our STEM image, and this was taken in our SEM, if you can believe that, we can clearly identify our gate region, diamond-shaped source drain that's typically made of silicon germanium, and our silicon fin region here. We use standard top-down sample preparation techniques to create our needle-shaped sample for Atom Probe, and we have 2 fins in our final needle-shaped sample. On the right over here, you can see our Atom Probe data obtained from this region. Here we can just make this transparent and do a little bit of an overlay to our electron microscopy images, and we find a close match of our 3D data set with our electron microscopy image. Now, once we have that and have identified all the elements, that's going to give us a really good understanding of the interplay between these different regions of the data set. Here in red, we have our SiGe region. In our orange ions here is our hafnium oxide. That's a high K dielectric. You can see here our silicon regions here in gray and our silicon oxide in light blue. So we can get a lot of different information from this device, and we have the ability to create 3D elemental data from all these structures. So let's take a look at that a little bit more closely, particularly within the SiGe source drain region. Here in our red ions we have our germanium, and our blue ions we have boron. Just by looking at this, we see that we might have a not uniform distribution of boron in this region. So how can we quantify that for Atom Probe? One thing that we can do is to create a concentration criteria that defines particular regions in this data set. So what I've done here is to create a 3% iso concentration surface. That means that inside these little blue regions we have a concentration greater than 3% boron, and on the outside we have less. And that will enable us to look at how the gradient changes as we go inside these high concentration regions and back into our silicon germanium matrix. What we find here is that as we go from the surface, which is 0.0 on our distance map, and go interiorly to the inside of these blue regions, we actually reach a concentration of up to 10% boron, which is extremely high for these type of structures. And then as we go towards the matrix, so that's back into our silicon germanium matrix, the average concentration of boron in these regions is only about 1%. And what is that ultimately telling us? At high concentrations, we observe quite a bit of clustering of boron. And if a dopant is clustered, it's not likely to be efficiently contributing charge carriers. That can just help us understand why we might not be getting the desired electrical behavior of the device, and it's really happening because of nanoscale changes. And lastly, I'd like to leave you with just a couple key takeaways from this webinar. First, that atom probe tomography is a materials analysis technique that can provide 3D elemental information at the nanoscale. And a lot of these can address problems that are of interest to the failure analysis community, such as concentration measurements, diffusion profiles, contamination, maybe chlorine and fluorine levels, clustering, and more. If you'd like to learn more, please reach out to us at atomprobe.com or CAMECA.com, and we'd be happy to answer any questions. Finally, I'd like to thank my colleagues at CAMECA for their hard work in all of these different areas. We've been working together for a long time, and it's really a joy to work here in Madison, Wisconsin. And I'd like to also thank you for attending this webinar and learning more about atom probe tomography. See you next time.